May 1993, a young Navajo marathon runner suddenly gasps for breath and collapses. Within hours, he is dead. Days later, at his funeral, his fiancée collapses with the same terrifying symptoms. She dies shortly after. That person who uncovered the truth about hantavirus and how it spreads. The Four Corners region had never seen anything like this before. A vast desert landscape where Arizona, New Mexico, Utah, and Colorado meet beneath an endless sky. To most people, it was quiet, remote, almost timeless. But beneath that silence, something invisible had already begun moving through the region. Something ancient, something deadly. Doctors were immediately confused. The victims were young, healthy, strong enough to suggest no obvious vulnerability, yet both had developed the same strange pattern: flu-like symptoms, fatigue, body aches, a mild fever, nothing that should have triggered panic. Then, without warning, their lungs began to fail. Breathing became rapid, shallow, painful, as if oxygen itself had become unreachable. Medical teams watched helplessly as healthy bodies collapsed in a matter of hours. There was no known explanation, no visible cause, no immediate connection. Only speed and death. Soon, hospitals across the region began noticing similar cases. More patients arrived with the same symptoms. Previously healthy individuals now struggling for air, their lungs rapidly filling with fluid, as though they were drowning from the inside out. Fear began spreading quickly. Rumors followed close behind. Some whispered about toxic exposure. Others feared an unknown epidemic. No one knew what they were dealing with. But one thing was becoming impossible to ignore. This was not random. Something was hunting silently through the Southwest, and no one yet understood how it was reaching its victims. Chapter 2: The Scientific Pursuit. Inside the laboratories of the Centers for Disease Control and Prevention, the atmosphere had changed completely. This was no longer a local medical mystery. It was now a national emergency unfolding under fluorescent lights and mounting pressure. Glassware clinked against stainless steel benches. Centrifuges hummed without rest. Computer monitors glowed through the night as scientists reviewed impossible data sets over and over again. Every hour that passed meant more uncertainty and potentially more deaths. The world was beginning to pay attention. An unknown illness was killing healthy young adults with terrifying speed. No clear source, no reliable treatment, and no confirmed explanation. Inside the CDC, investigators had entered what many later described as a medical manhunt, not for a person, but for a pathogen hiding somewhere beyond their understanding. The first suspects were obvious: plague, anthrax. Diseases already feared for their speed and lethality. Samples were rushed through testing pipelines. Lab technicians worked with mechanical precision. Pipettes moved from tray to tray. Protective gloves changed constantly. Specimens were isolated, tagged, and examined under controlled conditions. Then the first results arrived. Negative, not plague. More tests followed immediately. Anthrax screening began. Again, the same answer returned: negative. A wave of frustration moved quietly through the lab. Each eliminated possibility brought scientists closer to the truth, but also deeper into uncertainty. If it was not plague and not anthrax, then what exactly was tearing through these patients with such brutal efficiency? One scientist in particular became central to the investigation: Dr. Bruce Tempest, a virologist experienced in identifying dangerous viral pathogens. While others exhausted familiar possibilities, Tempest focused on a different theory. He suspected they were dealing with something viral, something not immediately recognizable through conventional screening. The challenge was brutal. Viruses leave behind tiny biological fingerprints, fragments so small they can disappear beneath layers of biological noise. Finding them was like searching for a single coded message buried inside static. That was when the investigation turned toward PCR technology, polymerase chain reaction, a method capable of amplifying microscopic genetic material until it became visible to analysis. In simple terms, it allowed scientists to copy tiny fragments of RNA millions of times, making the invisible visible. Lab teams prepared the samples carefully. Machines activated. Thermal cyclers began their programmed sequence. Temperatures rose, dropped, rose again, repeating in precise cycles like a machine slowly cracking a lock. Then came the moment everything changed. A signal appeared, small at first, but unmistakable. The code was there, hidden inside the patient samples all along. PCR had finally cracked it open. The unknown genetic sequence did not match plague, it did not match anthrax, and it did not belong to any common North American pathogen. Instead, the sequence pointed toward something unexpected, a hantavirus, but not the type scientists already knew. This was different, a previously unidentified strain, a new world hantavirus, a discovery both groundbreaking and deeply unsettling, because identifying the killer solved only half the mystery. The more urgent question remained unanswered. How was it spreading, and where exactly was it hiding? Chapter 3. The Source Uncovered. The answer was no longer hiding inside a laboratory. It was waiting somewhere beyond microscopes, databases, and sterile walls. To solve the mystery, scientists had to leave the world of machines behind and step directly into the landscape where the outbreak began. Under the scorching sun of the American Southwest, field teams spread across the Four Corners region. Miles of desert stretched endlessly beneath rust-colored cliffs and pale blue skies. But this was no ordinary wildlife survey. It was a hunt for an invisible killer. Researchers set thousands of live traps across remote home sites, abandoned sheds, barns, and open scrubland. Each morning, they returned to inspect what the desert had surrendered overnight. Rodent after rodent was collected, cataloged, and sampled. The search was methodical, exhausting, and increasingly unsettling, because a pattern was beginning to emerge. Again and again, one small animal kept appearing. Small body, large black eyes, soft brown fur with a pale underbelly, harmless at first glance, almost forgettable. Yet this species appeared everywhere investigators looked. Inside storage sheds, under floorboards, in closets, garages, cabins, and dusty attics above sleeping families. The animal had a name, the deer mouse, scientifically known as Paramyscus maniculatus, a common rodent native to North America, one that had been living alongside humans for generations. But blood analysis revealed something extraordinary. Many of these mice carried antibodies linked to the newly discovered hantavirus. Not sick themselves, not dying, just carrying it, acting as a natural reservoir. The realization changed everything. The killer had not arrived from some distant source. It had been living among people all along. But that raised another urgent question. Why now? Why had the outbreak exploded so suddenly in 1993? The answer traced back not to medicine, but to weather. Months earlier, unusually heavy rainfall had transformed the dry Southwest. Vegetation flourished, seeds multiplied. Piñon trees produced an unusually abundant crop of nuts. Food sources exploded across the ecosystem, and with food came reproduction. The deer mouse population surged dramatically. More mice meant more nesting, more droppings, more urine, and more opportunities for humans to encounter contaminated spaces. Invisible dust clouds carrying viral fragments upward. Humans inhaled them without warning, without taste, without smell, without awareness. Once inhaled, the virus entered deep into the lungs. There, it targeted the lining of tiny blood vessels. The immune system responded aggressively, too aggressively. A massive inflammatory reaction began. Blood vessels became leaky, fluid poured into lung tissue, and the body essentially flooded itself from within. A catastrophic immune overreaction known as a cytokine storm. The lungs filled rapidly, oxygen levels collapsed, and previously healthy people suddenly found themselves unable to breathe. The mystery was finally solved, not by finding a monster in the shadows, but by discovering something far more disturbing. The danger had always looked ordinary, small, silent, and living only a few feet away. Chapter 4: The Ancient Echo. As the scientific mystery finally came into focus, another truth quietly emerged alongside it. Long before laboratories, genome sequencing, or outbreak models existed, for many Navajo families living across the Four Corners region, mice were never seen as harmless background creatures. They carried meaning, warnings, stories passed carefully from one generation to the next. Among Navajo elders, there were long-standing teachings that discouraged close contact with mice and advised people to stay away from spaces contaminated by rodent activity. Old homes, storage areas, abandoned structures, places where droppings gathered unnoticed over time. These were not random superstitions. They were practical forms of survival encoded into oral tradition. A cultural memory shaped by centuries of observing the natural world closely. To outside scientists, this realization was deeply humbling. Modern medicine had used advanced molecular tools to identify the virus, but local knowledge had already recognized the risk long before the outbreak of 1993. In many ways, science had not replaced ancient wisdom. It had simply arrived at the same conclusion through a different path. That convergence became one of the most powerful lessons of the entire investigation. Knowledge does not always begin inside institutions. Sometimes it begins in stories, in habits repeated quietly across generations, in warnings that survive because they were earned through experience. Meanwhile, the medical community moved quickly to ensure the tragedy would not repeat itself unnoticed. Public health messaging changed almost immediately. Guidelines were introduced for safely cleaning rodent-infested areas. People were advised not to sweep or vacuum dried droppings directly. Instead, contaminated spaces had to be ventilated first and disinfected carefully to avoid disturbing infectious particles. These recommendations soon spread far beyond the American Southwest. Because hantaviruses were not limited to one region alone. Similar rodent-borne viral threats existed in multiple parts of the world. The Four Corners outbreak had exposed a vulnerability in how humanity understood emerging diseases. A healthy-looking environment could still conceal invisible biological threats, and an ordinary interaction with nature could trigger catastrophic consequences. The work of Dr. Bruce Tempesta and the investigative teams involved in the outbreak left a permanent mark on infectious disease research. Their efforts accelerated viral surveillance strategies across the globe. Hospitals became more alert to unusual respiratory patterns. Field ecology and epidemiology became more tightly connected, and scientists gained a deeper appreciation for how environmental changes can rapidly influence human health. But perhaps the most enduring legacy was psychological. The outbreak changed how people thought about disease itself, not always as something foreign or distant, but as something deeply woven into ecosystems we live beside every day. The Hantavirus was never a cinematic monster. It had no dramatic entrance, no visible warning signs, no obvious villain. Only a silent chain of events connecting rainfall, food supply, wildlife behavior, human routine, and biology. A reminder that the world is filled with patterns, whether we notice them or not. And sometimes, the most dangerous truths are not hidden in darkness. They are sitting quietly in plain sight, waiting for someone patient enough to finally understand them.